Home stretch of chapter 10, we pick back up talking about viruses because viruses are awesome. I love viruses, not because they get me sick or anything, but just the way they work is so totally crazy. So, viruses are a strange and amazing kind of life. You know how I always said the central dogma is DNA to RNA to protein, but I always specifically said cellular life. And that is because viruses are non-cellular life forms. And they are a unique form of life because they consist of a protein coat, genetic material, which could be DNA or RNA, and that's it. Sometimes they carry a few enzymes. Their protein coat has some other proteins attached to it to allow them to bind to cells, but honestly, that's it. Protein and genes. That's crazy compared to even the simplest prokaryote. A virus is incredibly simple. Viruses are also one of the few forms of life that are obligate parasites. They cannot survive without infecting a host cell. They don't reproduce outside of a cell. They really don't do anything outside of a cell. They just exist. And for some of them, for a very short period of time, if they can't get into a host. Viruses will die off, some of them very quickly, if they don't get access to a host. They need host cells to live. That's all there is to it. So, viruses have an external coat called a capsid. The capsid is a protein shell, a protective protein coat that contains the genome. There are 3 different capsid shapes in all the viral life forms. Icosahedral, which are 20-sided shapes. Helical, which is sort of a spiral tube shape, sort of like a slinky. And finally, complex, which looks like an Apollo moon lander. And I'll show you how they look like that in a little bit. There are, of course, some rare examples of things that don't fall into these 3 categories, like smallpox. Looks kind of like a big rectangle, but just about every virus falls into icosahedral, helical, or complex. And these complex viruses, this category, is exclusive to the bacteriophages. Remember at the beginning of chapter 10, when I talked about Oswald Avery and Hershey, well, not Oswald Avery, but the Hershey-Chase experiment that used bacteriophages? Well, all bacteriophages are these complex capsids that look kind of like an Apollo moon lander. So let's get into it. Some viruses have an envelope surrounding their capsid. Now, the envelope is a phospholipid bilayer with proteins embedded in it. And you might be saying to me, hey, dummy, we already learned that a phospholipid bilayer with embedded proteins and other stuff is a membrane. But guess what? Viruses are not a cell, so they don't have a cell membrane. They don't have a plasma membrane. In fact, they don't make their own envelopes. Their envelope is stolen from their host cell. Because when a virus replicates inside its host cell, it fills it up to the point that tens of thousands of viruses explode out of the host cell. And when they explode out of the host cell, they carry the envelope with them. So it's stolen membrane. And so we call it an envelope instead of a membrane because they're not making it themselves. They're not maintaining it. It's just there. And it's there to make them ninja viruses. So some icosahedral and helical viruses have envelopes. And that's a little piece of the host cell's membrane. And like I said, it ninjifies the virus because now the virus is carrying a piece of the host cell membrane that the immune system will identify as self. Now, it's not foolproof because there are markers on there that are viral proteins that the immune system can identify as viral in nature, but in general, that membrane makes it a lot easier for the virus to fly under the radar and infect other cells. So it's harder for the immune system to see the virus. Not impossible, harder. So here are the different viruses. So this is a helical capsid. You can see it looks kind of like Slinky. This is an icosahedral capsid. 20-sided looks kind of like a 20-sided dice. In fact, allow me to get one out because I'm a giant nerd and I keep 20-sided dice on me because I need them for Dungeons and Dragons. So, getting into my dice bag here because all respectable nerds carry a bag of dice because you never know when you're going to need to play Dungeons and Dragons. You may end up meeting a mystical wizard who tasks you with playing Dungeons and Dragons in order to save the universe. Right? Of course. Sure. Maybe. Leave me alone. Anyway, this is a 20-sided dice. So, there are 20 of these triangular segments throughout this structure. And that literally is the shape of the icosahedral virus. So this is a nice illustration of an icosahedral virus down here. And then over here is an enveloped helical virus. So this around here is the envelope. In fact, influenza, the flu, is an enveloped helical virus. So here's the helical, the helical capsid is inside here. So this isn't the greatest illustration of it, but here's the helical capsid. Influenza has an RNA genome. RNA as the genes, not DNA. This violates the central dogma. And this is why I always say the central dogma applies to cellular life. Because viral life forms don't always have DNA as their genetic material. It can be RNA. So the flu has RNA as its genetic material. And then it has this envelope stolen from its host. And then it has these little protein spikes, which the virus adds to the envelope during replication, so that it can bind to other host cells. And then this over here at the bottom right is our complex shape. So this is a bacteriophage, and it has an icosahedral head up here where the DNA is stored. Then it has attached to that a little neck. And then attached to that is a tail that's a helical tail that has a little base plate here and then tail fibers coming off of it. And these tail fibers are what attach to the bacterial host cell. And when the tail fibers attach, they bind onto it like this, and then they contract and send the tail like that and pierce through the bacterial cell wall. And once they pierce through the bacterial cell wall, it injects the DNA in the icosahedral head into the bacteria. Whereas the other viruses that prey on eukaryotes, like influenza, like other icosahedral viruses, these guys get into a cell by forcing the cell's membrane to endocytose them. Remember, endocytosis is where you have a cell membrane, and here comes something yummy, and the cell membrane sort of folds around it and then pulls it in, and it's inside the cell in a vesicle or a food vacuole. And so viruses actually force the membrane to close around them and stick the virus inside the cell, inside a vesicle or food vacuole. So Bacteriophages are crazy cool due to this unique structure. And all bacteriophages have a DNA genome, which makes it rather lucky that the Hershey-Chase experiment used bacteriophages, because if they used flu, it would have screwed everything up, because the flu genome is RNA. So hooray for the luck in that. So let's look at the different kinds of genomes a virus can have. So the genome is inside the capsid, all nice and protected. And it can be anywhere from as few as 4 genes to as many as 1,000 genes. So it's probably going to see something really big, like an extremely large virus, like smallpox, where you see close to 1,000 genes. Whereas 4 genes is going to be on something a lot simpler. So it's pretty crazy how viruses work. The largest genome is the pox virus. The pox virus is so big that it's almost visible in a light microscope, which is crazy. So the genome can be linear, kind of like eukaryotic genomes, or circular, like prokaryotic genomes. They don't care. It just depends on the virus. And then the genome can be RNA or DNA. Furthermore, the rules about the structure of DNA is always a double helix, RNA is single-stranded. Well, viruses say, screw your rules, man. I don't care. And they can have single-stranded DNA genomes and double-stranded RNA genomes. It gets crazy in there. So let's look at some of these crazy genomes. Viral genomes can be double-stranded DNA. Yawn. Just like regular genomes. Who cares? But then you can have single-stranded DNA, which is kind of weird, but still DNA, so I can see it. So once they get inside the host, they make a second strand and convert it to double-stranded DNA, so the host is like, oh, well, that I recognize. And then it reads the viral DNA and goes, oh, crap, I guess I'm just going to be hijacked and make everything the virus wants me to make. And then there's double-stranded RNA. What? Viruses are pretty close to the absolute only place you find double-stranded RNA. It's nuts. And so the virus has to bring with it its own enzyme, a viral polymerase, that can make DNA from its double-stranded RNA. And once it makes DNA from the double-stranded RNA, then the host can read the DNA and be hijacked by it. It can also have single-stranded RNA genomes. And that's not used to directly make proteins, because if you just come in with that alone, you only have one strand of RNA. That would be incredibly inefficient. So in order to be able to take that single strand of single-stranded RNA and make lots and lots of copies, you could have a viral polymerase make DNA from it, and then you may use the DNA to make copies of that single strand. Or you could even have a viral polymerase that makes RNA from the RNA. So it gets pretty crazy. In fact, if this were Bio 111, I'd be blowing your minds with the details. This is 105, so I'm not going to cram all of that down your head. So next is the funkiest genome of all, and that's the retroviruses. Retroviruses get pretty crazy, so hang on to your butts, people. We start with single-stranded RNA, and then we have a special enzyme that, in chronological order, takes the single-stranded RNA and makes single-stranded DNA. But it's not done yet, because then it makes double-stranded DNA from the single-stranded DNA. And then it does something absolutely nuts. And that's it sticks its double-stranded viral DNA into the host's DNA so that infected cells now permanently have viral DNA in them. HIV is an example of a retrovirus, and this is one of the reasons why HIV is so hard to cure, because it becomes part of your genome. And so you can't just inject something that attacks the genome, because it'll attack your whole genome. That's pretty scary. HIV is even scarier because it happens to integrate itself into the genome of your white blood cells. And it's those white blood cells that are responsible for attacking viruses and other pathogens. And when that virus wants to, it will shut down your white blood cells and essentially make you have zero immune system. And that's why HIV can turn into AIDS, which is an autoimmune deficiency syndrome. And so basically when you get full-blown AIDS, your immune system craps out on you, and then you don't die from your HIV infection. You don't die from AIDS. Rather, you die from the hundreds of other pathogens that you can no longer defend yourself from. So, pretty funky, kind of terrifying. Let's look at the basic life cycle of a virus. The virus enters the host cell. Then its capsid opens and it releases its genome into the host. And then the host reads the viral genome and makes a few proteins that then shut down the host's own functions. The host will stop reading its own DNA and start reading the viral genome. And then host or viral enzymes produce mRNA and start making viral proteins. And then capsid proteins and viral enzymes are produced. In an enveloped virus, something else happens. And that's that viral glycoproteins are embedded in the host's membrane. These viral glycoproteins allow an enveloped virus to force a cell to pull it in. So that's why it transfers viral glycoproteins to the host membrane. And where those glycoproteins end up is where those enveloped viruses are going to erupt out of the cell. That's where they're going to steal membrane pieces so that they can have those viral glycoproteins to allow them to infect other cells. So once we've produced capsid proteins and viral enzymes and prepared the membrane, if we're enveloped, to allow the viruses to explode out, it's time to make copies of the viral genome. And so then the host's own enzymes crank out copies of the viral genome. And once you have genome, ingredients for the capsid, viral enzymes, and the membrane primed for the virus, you start to put everything together. And that's pretty cool. The genome and the enzymes aggregate, which means they sort of come together into the same spot, and then the capsid forms around them. And this is really neat because the capsid, the proteins are attracted to each other just kind of on their own. And so with just a little bit of help from the cell, the capsid chunks come together and wrap around the genome and the enzymes, and that virus is now ready to explode out of the host and go infect other cells. So it exits the cell. And with a large number of viruses, when they exit the cell, they blow apart the cell's membrane and kill the cell. So that is right. A viral infection is literally killing you from the inside. That is, again, terrifying. So When you get a viral infection, it becomes a war between your immune system and the virus, in which the virus starts blowing apart your own cells, killing cells left and right. The horror! And then the immune system rolls up and starts killing the viruses. And then you got cells being killed, and then the immune system killing the viruses until the immune system kills enough viruses that they can no longer keep your infection going. And then your immune system mops them all up and you become virus-free. Unless, of course, it's chickenpox. You don't need to know this about chickenpox, but chickenpox is a class of viruses you might know. It's a herpes virus, so that's pretty scary. Anyway, chickenpox, when it infects you, it gets into your nerves, and it goes down to the places right by your spine, and it hangs out forever. You can never fully get rid of chickenpox. Of course, now there's a vaccine, so you spoiled, rotten kids can keep yourselves from getting chickenpox. Yeah, I was born much later than you, Jonathan, so I don't have to get chickenpox. Well, screw you. I have chickenpox near my spine, and I'm going to have it forever. And when I'm old, it can reemerge and cause shingles, and that sucks. So I don't know what I was getting at there, but viruses are terrifying and so cool. So when they exit the cell, they usually lyse the cell. They usually break that membrane to pieces. Naked viruses will break through the membrane in order to lyse it. They leave giant holes in the membrane where they bust it out. Enveloped viruses, they exocytose. In other words, they break out at the part of the membrane that has embedded glycoproteins. And they take that piece of envelope with them, which generally lyses the cell because they're stealing the cell's membrane. And so, again, they're putting holes in the cell. There are cases in which when the virus exits the cell, it doesn't kill it. For instance, HIV, when HIV decides to replicate and infect the rest of your immune cells, it's not killing your immune cells, which allows it to hang out and then crank out AIDS and destroy you at a later time. So, bacteriophages are pretty funky. They have a neat life cycle. Bacteriophages have a two-step life cycle. There's one possible life cycle called the Lytic Stage, and a second possible life cycle called the Lysogenic Stage. So we'll look at life cycle 1, and that's the Lytic Stage. And in life cycle 1, the bacteriophage binds to the bacteria, the tail sheath pierces the membrane, kind of like a hypodermic needle, and then the viral DNA is injected into the bacterial cell, and the cell's own DNA is literally destroyed, leaving only viral DNA behind. At which point, the virus synthesizes all its part and assembles together, making new viruses, and then blows apart the host cell as all the new phages are released. So the lytic stage kills the host cell, destroys its DNA, and releases everything. The lysogenic stage is where the phage injects its DNA as normal, but instead of destroying the host's own DNA and then making viruses and blowing the host apart, it will integrate its DNA into the host's genome, similar to how HIV works. That DNA integrates into the host genome, and then the host is stuck with that DNA as part of its genome. And you know what? This is kind of neat. And when I say kind of neat, it can produce thoroughly unexpected things. You see, scarlet fever is caused by a bacteria that has been infected with a bacteriophage. And that bacteriophage just happens to carry a gene that allows the bacteria to cause the scarlet fever disease. A bacteria of that species that hasn't been infected cannot cause scarlet fever. What? It's crazy awesome. You don't need to know that about scarlet fever for the exam. It's just neat. Now, once the DNA is integrated into the host genome, binary fission occurs as normal. And so your cell goes about its business just as if it weren't infected and replicates itself. Because remember, binary fission is what prokaryotes do instead of mitosis. Mitosis is for eukaryotes. Binary fission is for prokaryotes. And that's where they double. So our infected cell becomes two infected cells, which becomes 4, 8, 16, 32, 64, 128, 256, 512, 1024, 2048, 4096, 8194, or 96. Yeah, and so on. So you get multiple daughter cells, and that DNA integrated into that genome is now called a prophage. So you have a bunch of daughter cells with that viral DNA as part of their genome, that prophage as part of their genome, and it just continues over and over and over. Except that at any point, that prophage may just exit the genome. And when it randomly exits the genome, it will start a lytic stage. So it makes lots and lots of cells with viral DNA, and then the viral DNA can exit and start a lytic stage. So here we go. On the left, we have a lytic stage. On the right, we have a lysogenic stage. So here comes our bacteriophage attaching, adding its genome. And then here we go. If we're the lytic stage, we destroy the host genome, and we start making viral copies. And then we release the viral copies, blowing apart our bacterial host and killing it, at which point these little copies go and attack other cells. Or, when it attaches and injects its DNA, it might just integrate its DNA into the host genome and then have binary fission producing doubles, and it just continues over and over again, except that every once in a while, that host genome may pop out and start a lytic stage. So it's pretty useful. If a bacteriophage happens to infect a bacterial cell in an area where there's very, very few bacterial cells, it may go into a lysogenic phase, where it holds off on killing the bacteria until there's enough that it would make it worthwhile. So, kind of pretty awesome. How do you protect yourself from viral infections? Well, there's your own immune system. When your immune system detects a virus, it will get sensitized to that virus and start attacking that virus. So getting your normal immune response means getting the virus and then developing immunity to it, because your immune system responds and starts destroying it. This is kind of useful because once you get the virus and fight it off, you produce memory cells. You don't need to know the term memory cells, but these memory cells then can, if you ever get the virus again, start cranking out antibodies to fight the virus. And it allows you to respond to a subsequent viral infection much, much faster. So it sort of protects yourself. So you fight off the virus, and then you get antibodies that can come back if you ever get infected again. So your immune cells remember the virus, and they can fight it off much faster later on. Then there's vaccines. With vaccines, instead of getting the viral infection, you generally get infected with pieces of the virus. So either a heat-killed virus that can't cause disease, or little viral proteins or other viral molecules. Your immune system recognizes these as foreign things that must be destroyed. What's useful about a vaccine is that you're not getting the disease, but your immune system still recognizes these things as foreign and starts to remember them. And these things can be remembered if you ever get the actual virus. So it sort of primes your immune system without the annoyance of actually getting the disease. So vaccines are incredibly awesome. Between sanitation, learning about viruses, and vaccines, we have been able to drastically reduce a lot of the persistent viruses that have plagued mankind. For instance, smallpox does not exist anymore. We completely eradicated smallpox, which is pretty awesome. Polio in the US is virtually non-existent because of increased sanitation practices, better knowledge of viruses, and vaccines. Vaccines played a big role in reducing polio infections. So vaccines are, in a word, awesome. So if you ever hear someone who's all like, vaccines cause autism and they make you sick and blah, 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 go ahead and, offer to have them unvaccinated and exposed to polio. See how they feel about that one. See that, because there's absolutely no scientific link to autism. Zero. The one scientist who found the scientific link turned out to end up getting paid extra money because he got kickbacks, because the company he was funded by had an interest in there being a scientific link between vaccines and autism. And furthermore, when people reviewed his actual research, it turned out he just went ahead and made up his data so that it supported vaccines causing autism. And as it turns out, no, they don't. Sorry, anti-vaccination crowd. So those people are nuts. Anyway, end rant. You don't have to know my giant rant on anti-vaccination. Vaccines have the bonus of being non-pathogenic. But wait, you say, I've gotten sick from vaccines. In fact, you have not. One of the things that often blows people's minds is that your immune system itself causes reactions that may make you think you're being sick. Your immune system, in fighting diseases, will cause fevers. It can cause swelling. It can cause inflammation. So some of the things we associate with being sick is actually your natural immune response. Not fun, but you're not actually getting the virus. You are not getting sick. That virus is dead. It may even be just little chunks of the virus. You cannot get sick from it unless it is what they refer to as, and you don't have to know this, but a live attenuated virus. Live attenuated viruses are often used in people who have allergies to the ingredients in heat-killed vaccines or viral protein or other chunky vaccines. And they're essentially living viruses that have been sort of cultured to be non-pathogenic. And if you're unlucky, you can get sick from one of those. But read up on what vaccine you're getting. If you're not allergic, you can probably get a dead virus or chunky virus vaccine. And it's impossible to get sick from those. So, it's non-pathogenic, and it still stimulates the immune response, and it still allows the immune system to remember those components. And if you ever get the live virus, your immune system will say, hey, I remember these pieces, and attack the virus. So, pretty useful. All right. Acquiring immune responses, like I said, you can be exposed to the virus itself. You can actually be exposed to a closely related virus. One of the interesting examples of this is the Spanish flu, which I think rolled through the human population sometime around, I think, 1919, but don't quote me on that because I'm just trying to remember that from just offhand. Anyway, the Spanish flu rolled around and primarily killed young men. Older men turned out to have resistance to the Spanish flu. They weren't really killed in too large numbers by the Spanish flu. And the reason for that is that earlier in the late 1800s, a closely related flu epidemic had rolled through. And all the people who lived through that flu epidemic ended up having an immune response that could react to the Spanish flu and fight it off. So even if you're not exposed to that specific virus, if you're exposed to something closely related, you can fight off that specific virus. Kind of neat. So cowpox, very closely related to smallpox. In fact, the very first vaccine ever developed by humans was cowpox. People were injected with cowpox, and it allowed their immune system to develop antibodies to cowpox, which could then attack smallpox. And that's how we eradicated smallpox. The vaccine for smallpox used all the way through the late 50s and the 60s, when smallpox was eradicated, was the cowpox-based vaccine. Pretty neat. In fact, one of the neat things about the discovery of cowpox as a vaccine, if anyone's ever sort of acquainted with old sort of European fairy tales, one of the themes you often see is sort of the beautiful milkmaid. And what's neat about that is that that's actually based in fact. Milkmaids were women who obviously milked cows. And in the course of that, they were often exposed to cowpox. And they were some of the only people exposed to cowpox. And so they had a natural resistance to smallpox. Smallpox causes disfiguring pustules on you and leaves ugly scars behind. And so these milkmaids who were resistant remained beautiful. And so that's why you see it in some sort of European fairy tales. The beautiful milkmaid. She had resistance to smallpox due to being exposed to cowpox. Neat. And there's my Spanish flu. So, that's cool. All right. We're getting close. We're getting close. There's viruses in plants. Plants have a thick cellulose cell wall. So viruses can't explode out of the cell wall. This is kind of a problem because that's sort of what viruses naturally do. They blow up out of the cell and then they go infect other cells. However, plant cells happen to have little canals between all of them so that they can share nutrients that come up from the roots. And viruses will exploit these canals. And so they get to other healthy cells by traveling through the canals. And we call that horizontal transmission. And it requires often the virus will get into the plant in the first place by exploiting a broken or damaged plant. It will get into cells that have been damaged. Oftentimes, plant viruses will hide in insect salivary glands. It's one of those things that's details you don't need, but that are just really cool. They will hide in insect salivary glands, and they don't cause sickness in those insects. But when the insects come along and start eating leaves, they break the cell walls of those leaves. And the viruses are spit out in the saliva, and then they can invade the broken cells, and then they can infect them and then start moving through those canals. So they can be carried by wind as well. And like I said, insect vectors and often animal vectors can carry plant viruses. And we call those living organisms that carry viruses live vectors. So once they get inside the plant, they travel between cells. Another way viruses replicate in plants is vertical transmission. In vertical transmission, the virus not only infects regular cells, but it will infect the sex cell. And so the virus can infect a sperm or an egg. And then when that sperm and egg are fertilized to produce a seed, that seed is infected. And so it will automatically grow an infected plant. So it infects gamete-producing cells, which produces infected gametes, which then produce infected seeds. And if you've ever seen the pretty tulips with the white stripes in the flowers, those are actually seeds that were infected. And that is a virally infected flower. So, pretty neat. Okay, last couple of slides in which we talk about the kind of scary stuff. Things that we don't really have a defense against. The first thing is the viroid. The viroid sounds kind of like some sort of evil alien in a science fiction novel. You know, like, Zap! Brannigan! We need to destroy the viroids. They're invading. Don't worry, I have my laser pistol. You know, that kind of thing. But, in fact, viroids are not scary aliens. They are actually circular RNA. And that's it. There's no protein coat. There's no accompanying enzymes. It's just a little chunk of circular RNA. And that little chunk of circular RNA just happens to be infectious, and it will cause disease. And those diseases are lethal. They kill. And there's not much humans can do against them. However, luckily enough, these viroids only infect plants. So, oh, good. Whew! They're not killing us. They're only killing plants. Of course, that sucks if there is a viroid that evolves that attacks like major crop plants, and there's nothing we can do about it. But won't be directly killing us. Hooray! Okay, well, since that ended on a nice note in which you're like, oh, phew, let's go ahead and terrify you with prions. Prions are a protein, not even RNA or DNA. Protein. A protein that became randomly misfolded. In fact, every single one of you, me included, all humans carry a gene, all animals really, they carry a gene called the prion protein gene. And it's actually a normal part of your life. That prion protein gene codes for a protein that is vital to your being alive, does important things. However, that prion protein you are making all the time could randomly become misfolded. Who cares, you say? Well, it's just a protein, but it's infectious. What does that mean, you ask? Well, when it touches any other kind of protein, it causes that protein to automatically misfold into a prion. And it essentially causes any protein in your body that just comes into contact with a prion to become a prion. And so you start converting all your proteins into prions. Why is that horrifying? Because pretty soon those prions accumulate and they start blocking off the blood cells in your brain, and then it starts killing brain cells and your brain becomes Swiss cheese. So, anytime any other protein touches a prion, it automatically misfolds. The prions start building up in your blood and they block blood flow, turn your brain to Swiss cheese, and kill you. Universally lethal. And if you get a prion disease, there is no cure. One prion in your body, and you're gonna die. Mad cow disease is an example of a cow prion disease. It's in cows. However, remember how I said a prion protein that hits any other protein causes it to become a prion? if you eat cow meat that happens to have a prion in it, you get the prion disease and you're gonna die. These prions cannot be denatured. You have to literally turn your steak into a black carbon chunk in order to kill the prion. If you get the prion disorder, nothing short of setting yourself on fire will stop the prion disease. And that kind of defeats the purpose, because then you die in flames, which is probably less pleasant than having your brain turned to Swiss cheese. There's no known way to stop or fight the infection. And if you get a prion, you dead. So, hooray! So that's pretty horrifying. So let's go ahead and end this lecture on a lighter note, okay? Ooh, look at the cute bunny. It's so adorable. Oh, his little kitten's hugging. Oh, look at the puppy. Oh, little bitty ferret sleeping in a little heart shape. Oh, now you feel better. You can stop rocking back and forth in the fetal position. Good luck on the exam.